Revision video with Mr. Phillips, and today we are going to be looking at hormones and the control of fertility and their role in the development. And this sort of splits into 4 stages: IVF treatment, embryo development, pregnancy control, and then a very small introduction to hormone replacement therapy. And just to remind you, this is not enough material to learn just off on its own. You must be doing extra revision and work to support your studies. Start off with IVF treatment, which stands for in vitro fertilization. Start off, to understand the process of in vitro fertilization, we need to understand the hormones involved in the menstrual cycle. And we talked about these in the previous video, but just to remind you, there is follicle stimulating hormone, which is a protein hormone made in the pituitary gland, which binds to the receptors on the follicle cells in the ovaries, and it starts the eggs to be matured in the ovaries. Estradiol, which is a steroid hormone made in the by the follicle in the ovaries. That then impacts the gene expression on uterus development, so it causes the uterus lining to become thicker in preparation for the acceptance of a luteinizing hormone. Then is a protein that is made by the pituitary gland. It binds again to the follicle cells in the ovaries, which then stimulates ovulation on day 4. Is a steroid hormone made by the follicle cells and the corpus luteum after the egg has been released, after the follicle has been released, I should say, that influences again gene expression on the uterus, and it maintains that uterus lining to the end of the cycle or longer if pregnancy occurs. So IVF, then, in vitro fertilization, is a complex procedure, okay, and it differs on who is doing it and the needs of the patients. So what we're about to discuss is the general procedure as a result. So obviously it's tailored for the different patients. So the first thing that would happen is that the woman would inject drugs to stop the production of FSH or LH. This is done because if you stop the production of FSH, FSH then stimulates the production of estradiol and progesterone, suspending the normal menstrual cycle, and that allows doctors to control timings and the amount of egg production. Okay, so in order to do this, they inhibit the pituitary gland. So again, they prevent the production of SH or LH, and that is to stop the production of estradiol and progesterone, which are triggered by their production, and that means they can control the timings of egg production, etc., for this process. And the pituitary gland is the gland where it all starts, so you have to inhibit that one, which stops the release of the FSH or the LH. You then have something called intramuscular injections of FSH given for 7 to 12 days. Generate high follicle stimulating hormone, more than normal, to allow more than the normal amount of follicles to develop. So once we've got the timing ready, we inject this high number for a high number of days. Ideally, you'll have 8 to 15 follicles stimulated, which will mean that each one of those will have an egg, which increases your chance of being successful. And this is obviously stimulating the follicles in the ovaries. So just to recap, for the first stage, we prevented the production of FSH and LH to control the exact timings. Then step 2 is when we're ready to cause the egg to be matured, we inject them with FSH for 7 to 12 days, ideally stimulating up to 8 to 15 follicles to mature with an egg in there. And that is obviously the ovaries where the follicles mature. Then inject something called human chorionic gonadotrophin hormone to the follicles when they are 18 millimeters in. This is normally released from a 1-week-old embryo to tell the mother it needs sustenance from the endometrium, which is the lining of the uterus, and this means the uterus lining will start then preparing for implantation. So we're making the female's body, the endometrium lining, think that it's going to have an egg which is going to be accepted and implanted. So it's preparing for the egg to be implanted so it can start to create the nutrients needed to maintain a uterus. The eggs will then be collected 35 hours after the HCG injection, which is a minor surgical procedure taking about 20 minutes, and a micropipette is mounted on an ultrasound scanner and is passed through the uterus wall to draw the eggs out of the follicle. Then is mixed with up to 100,000 sperm in sterile conditions at 37 degrees and left for 24 hours to allow fertilization to happen. We're obviously used there, and that is to increase the chance of fertilization. If fertilization occurs, the embryos will be isolated and they'll be placed into the uterus at 48 hours old. The female is given extra progesterone to ensure the uterus lining is maintained. So that is how IVF works. Now let's have a look at pregnancy testing. So hormones are also involved in the test for pregnancy, and that is due to the fact that this hormone here, HCG, human chorionic gonadotrophin hormone, levels increase during pregnancy, as I said a few moments ago, to develop the lining of the uterus. So the embryo releases human gonadotrophin hormone, and it actually releases it directly from the blastocyst stage onwards. It's vital for the first 10 weeks as it causes the corpus luteum development and the progesterone secretion, which breaks down the uterus lining, prevents the breakdown of the uterus lining, I should say. After 8 to 12 weeks, the placenta secretes progesterone, and cells in it, which are called the trophoblast, continue to secrete HCG hormone for the rest of pregnancy, continuing the release of progesterone and maintaining the endometrium, so the uterus lining, as a result. Pregnancy tests work by detecting the release of HCG in urine. This earlier on in school to some level. So pregnancy tests work detecting the HCG and using something called monoclonal antibodies. So if you are pregnant, you will be asked to urinate on a pregnancy stick test, and that contains HCG hormone. So this hormone is a small hormone that can come out via your kidneys, via your urine, and again, you only produce this if you are female and pregnant, although males can also produce it as a result of testicular cancer, so it's used as a cancer checker for males. HCG then binds with what we call monoclonal antibodies, and those monoclonal antibodies here, which you can see at number 2 here, are all unique, and they all have a receptor with a shape on it that binds to the HCG hormone, and they will be have a blue dye attached to them, which eventually is what you see appear on the stick. Now, at the test site then, if you are pregnant and you do have this HCG hormone, there is a layer of immobilized monoclonal antibodies. So these red ones here are monoclonal. They are attached to this position in the tester kit. And what will happen is the HCG will also bind to them in the red area here. So they will bind along this bit here, okay, and therefore they will bind to the immobilized antibodies, and therefore also the antibodies, the monoclonal antibodies with the blue dye, will also attach. And if that happens, because you are pregnant and you have the HCG hormone, you'll get the first blue line in the test site. Some of the monoclonal antibodies will not have HCG. There's too many of them to all have HCG hormone. So these ones will move past. They won't attach to this antibody, which is complementary to the HCG. They will move further down the stick, and they directly bind to another immobilized antibody, which is complementary to their shape, and therefore you get a second line as a result. Now, if you're not pregnant, obviously you have no HCG, therefore no HCG will bind to these immobilized antibodies here, so you don't get a line, and then all your antibodies will attach to the second set of immobilized antibodies, so you get a control line there to show the sticks worked, but you are not pregnant. And if you're not pregnant, only the control line forms. So what is the role of hormones then further on? So once you are pregnant and you have a embryo attached to the endometrial line. Have a look then. So the zygote, when it is formed, has two haploid nuclei. So if you think about it, you have a nucleus from the egg and a nucleus from the sperm at first. 6 hours into fertilization, DNA replication takes place, and in each of those nuclei, and it takes about 3 hours to complete. The DNA then undergoes major changes in the pattern of gene expression, and by 30 hours, they divide jointly by mitosis, producing two genetically diploid cells, which then undergo cell division as normal. So effectively, they take a bit of time to fuse together and form one nucleus, and then from then on, it becomes diploid mitosis. So the next few days then, the embryo undergoes rapid rounds of cell divisions and cell cycles, with the number of cells doubling every about 18 hours. Remember, this is for humans. Cells get smaller as growth doesn't happen between each stage, so the cytoplasm is divided from the large initial zygote. So you see here, you start off with one big cell, and then it goes to smaller cells, half the size, and then a quarter of the size, and then an eighth of the size, and a 16th of the size, and eventually we get something called a blastocyst. And this is when the embryo moves from a solid ball of cells to a hollow ball created by unequal cell divisions and cell migration, making the blastocyst. And this is the first point where differentiation. We're at the blastocyst, we need to understand, well, what happens to that to make it become implanted into the embryo. So the blastocyst has about 250 cells, as we said before. They are unequally distributed now, so you've got this sort of hollow, not hollow, but liquidy center called the blastocele. Okay, you've got an inner mass of cells here. The outer layer of cells are called the trophoblast, and there's a tough layer called the zona pellucida outside of the blastocyst. Remember, you should remember those, but that worm zona pellucida from the original egg, because don't forget, this is the egg that has now had several divisions happening in it after the fertilization. Obviously, it became the zygote. Now, it will gently then migrate over due to gentle wafting of the cilia in the oviduct wall, which slowly moves the blastocyst towards the uterus. And then when it reaches the uterus, there has to be a key change in that the tough zona pellucida, which protects the embryo until this point, has to now break down, because these inner cells have to combine with the uterus. So what has to happen is that at this point, the food supply is going to be starting to run low. So the blastocyst has to now conjoin with via what we call implantation via the wall of the uterus, known as the endometrium. So the food reserves are used up, and we have a limited amount of time. So the outer layer of the blastocyst, which we call the trophoblast, develops finger-like projections that grow into the endometrium. And then this allows exchanges with materials of the mothers, including oxygen and food, to allow the growth of the blastocyst. So if we continue, we get to 8 weeks old, and by 8 weeks old, the blastocyst inner mass starts to develop into the human body, and it has started to form bone and tissue, and now should be called a fetus, no longer an embryo. The individual will soon be recognized as male or female. So you see the rapid development once it's implanted into the mother's uterus and it starts to get oxygen and food. And then if we look at how this sort of works quite quickly here, just I found this as a nice summary picture. At week 4, the heart starts to beat, the arms start to appear, the liver, the pancreas, the gallbladder forms, the spleen forms. So you see differentiation happening. Eyes, legs appear week 5. The blood begins to circulate. Week 6, the lungs start to form, fingers and toes start to form, and hair follicles at week 7. Elbows and toes become visible, and then the face starts to look more human, and you start to get ears forming at If we look at the first 9 months, and more importantly, how hormones contribute to the development of the first 9 months, is another area that is quite important, that will lead to the birth of the child as well. So just to make sure you're happy with the placenta and what its role is, humans are placental animals. So we are placental mammals, I should say. So here's some examples there for you. There are different mammals. Not all mammals have placenta. So monotrines are mammals that lay eggs, such as the duck-billed platypus, and marsupials give birth to relatively undeveloped undeveloped offspring, which then develop in the pouch. So you can think of like the kangaroos, etc. The placenta is important because it means the human fetus can remain in the uterus for months longer than marsupials, and the placenta then is fetal tissue in intimate contact with the maternal tissue. So basically, it allows the exchange of materials from the fetus to the mother, allowing the growth of the fetus. And you can see that because of placentas, we have a longer time in what we call gestation, how long you're pregnant for. It means that we can remain in the uterus for months longer than marsupials, and that is obviously quite important. So here you can see the sort of setup. You have the umbilical cord, which you'll be familiar with, which ends in the placenta, and it's this really thin layer here, which is magnified here, that allows the exchange of materials that we'll look at now. So the placenta is fetal tissue in intimate contact with the maternal tissue in the mother's uterus wall. So the placenta is part of the fetus. The fetus develops a membrane to become an embryonic sac which contains amniotic fluid, and that supports the baby and protects the fetus. So you have this membrane all the way around the baby called the amniotic sac, and in there's amniotic fluid, which stops if the baby gets banged or the mother falls over, for example. It acts as a cushion to protect the. So the basic unit of the fetus is called the placenta villus, which you can see are these bits here. Now we talked about this a few minutes ago, that the blastocysts made finger-like projections into the endometrium lining of the uterus, and that is what these are here. So their numbers increase during pregnancy constantly to meet more and more demand. So if you think about these, think about them almost like external gills is what I would say. So if you think about like an axolotl which has external gills, these have a large surface area, a good blood supply. They're 1 cell thick, which allows materials such as oxygen and nutrients to pass from the mother's blood into the blood of the developing fetus. The maternal blood flows around the intervillous space, so it leaves the mother's blood vessels. So here you see what happens is you've got the mother's blood vessels here, so the blood actually enters this space around the placental villus, which means there's a good amount of exchange happening directly with the baby's tissues. And the fetal blood flows in the capillaries into the villus. So you see the fetal blood comes in here through these blue narrow tubules and then back here in capillaries in the villus. So there's a really close exchange of 1 cell between the mother and only 5 micrometers, in fact, is the distance between both types of blood which aid exchange. So that is an incredibly small distance for diffusion of materials to happen through. And the baby obviously needs to get things in, such as oxygen, water, nutrients, hormones, antibodies to protect against disease, drugs and viruses out of its body. Whereas from the baby, drugs and viruses will go into the body that we've taken from the mother, but the baby needs to get rid of carbon dioxide, water, excess water, urea, other waste products, and hormones in excess. And that 5 micrometers is extremely thin layer, allowing that to happen. The cells separating both are called the placental barrier and are selectively permeable, allowing some but not all substances to pass. So I said earlier, things like carbon dioxide, oxygen, and urea pass by simple diffusion, facilitated diffusion with glucose and amino acids, osmosis, obviously water, and then there are extra processes called endocytosis, which allow antibodies to move changes happens around 9 weeks, and this can be one of the common causes of a miscarriage if this goes wrong. So what happens is the placenta starts to secrete estradiol and progesterone in large enough amounts at that point to maintain the lining of the uterus in that particular area. So the corpus luteum, which if you remember we said at the start of the video, was releasing these hormones to allow the embryo to get ready to implant into the uterus lining, is no longer needed. Now, if this failures to switch over, this can cause a common cause for miscarriage, because too much of the uterus will be adapting and it won't be focused on the placental area. Now, progesterone then is important because it inhibits the secretion of oxytocin by the pituitary gland, and this prevents the outer uterus muscular wall from contracting. So you need to have a constant level of progesterone during pregnancy. That means then obviously something must happen at the end of pregnancy, which prevents all this. So at the end of pregnancy, the placenta stops releasing progesterone, and then, as we said a moment ago, that means the hormone oxytocin can then be released by the pituitary gland. Oxytocin stimulates the myometrium muscle contractions in the uterus, and that then is what we would start to call contractions, and these are then detected by stretch receptors, which stimulate more oxytocin production from the pituitary gland, which means more oxytocin is released, which leads to more vigorous contractions, triggering more oxytocin to be further released. And this continues then until the cervix opens and the baby is born. So it's a rare benefit of positive feedback. Normally positive feedback causes health issues, but this one is an example of where it has a positive effect. So if we look at the picture on this one, just to make sure we're happy with it, the picture shows that if we start here, oxytocin, so remember we said the placenta stops releasing progesterone, and oxytocin therefore is released. That causes the muscle lining the uterus, called the myometrium muscle, to start contractions. That causes then the stretch receptors to be pushed, which increases the amount of oxytocin release, which increases further the amount of muscle contractions happening, and more then the baby can move, which generates even more stretch receptors, which triggers more oxytocin to be released in positive feedback until the baby is. As contractions occur, what happens is the cervix relaxes, and the muscles relax, causing the cervix to dilate, which means open. Now the cervix has been holding the baby, and so here is the cervix inside the reproductive organs here. It's been holding the baby in position as it's grown for the last few months, particularly the later months of pregnancy. And then what will happen is, due to the contractions, the amniotic sac, which if you remember had the amniotic fluid protecting the baby in, that will actually burst as a result, and that is when we say the waters burst in pregnancy, which means you know you're about to give birth. Further uterus contractions then push the baby out of the cervix and out of the vagina, so the baby is then born and the umbilical cord is broken, and that then leads to the baby automatically to take its first breath. One thing obviously here which you just need to be aware of, I think, is that obviously hormones are a key part of the developmental stage, and obviously for many women at later on in life, when you hit the menopause, hormones may cause some issues, and therefore you might look for hormone replacement therapy. So it's used to relieve the menopause symptoms by supplementing women's estrogen and progesterone that falls as you approach the menopause. It can also prevent conditions which can lead to this more common after menopause, such as osteoporosis, which is a weakening of the bones. However, there is some suggestion that there are risks of hormone replacement therapy for other diseases, including coronary heart disease. So this is an area where you'd have to look at data and work out, is it worth taking them? Do the benefits outweigh the side effects? And it's a constant area being researched and reviewed by health authorities.